Now that we've looked at metabolism, let's talk about how bacteria grow. Why do we care how they grow? Well, one, we got to be able to grow and to study them and to see what they are. And two, if we know how they grow, it's easier to figure out how to stop them from growing or how to kill them, right? Heat, cold, gases, pH, radiation, hydrostatic pressure, osmotic pressure, all these things can affect, and other microbes as well, can affect how a microbe grows and survives. So microbial cells cannot regulate their own temperature, and so they're kind of at the mercy of their habitat. The range of temperatures for the growth of a microbe can be expressed in three different what we call cardinal temperatures, okay? And that's the minimum and maximum temperatures that define the limit for growth and development of an organism, okay? The range of temperatures for the growth of a given microbial species is going to be this. We've got our minimum temperature, and that's the lowest temperature that allows a microbe's continued growth and to continue its metabolic processes. Below this temperature, it's going to its activities are going to be limited. Okay. They're going to stop basically. At the maximum temperature, this is the highest temperature at which growth and metabolism can occur before the proteins are denatured. Okay, I'm going to explain what denaturation is. The optimum temperature, and of course, if you have denatured proteins, they're not working, and so again, you're going to stop your metabolic processes. The optimum temperature, again, that's our happy place, right? This is the intermediate between the minimum and the maximum that allows the fastest rate of growth and the fastest rate of metabolic processes to occur. Some microbes have a very narrow temperature range, and some are more generalist, so they have a broader range. And so we're going to classify microbes into ecological groups based on whether or not they grow best in cold, moderate, or hot temperatures. All right, so here are these ecological groups here. We've got psychrophile, psychotroph, mesophile, thermophile, extremophile. So phile means loving, okay? All right, so a psychrophile, that's going to be a little blue line here. This is the optimum is going to be at the peak, okay? So the optimum temperature here is going to be between 0 to about 15. It looks more like 10 here to me, but generally about 15 degrees Celsius, and so they're capable of growth at 0. They're going to be obligates when we look at cold, and they cannot grow above 20 degrees Celsius, so they have to be in a cold area. All right. Storage at a refrigerator temperature would actually encourage them to grow rather than inhibiting them to grow. Okay. Natural habitats for psychrophilic bacteria like fungi and algae are going to be things like lakes, rivers, snowfields, obviously, since that's very cold, polar ice caps, and the deep ocean. Now, we used to actually believe, in fact, I was taught that things did not live in areas like the polar ice caps or deep sea thermal vents, but we have, or in the high salt areas, have discovered that that is not true, and that we actually have living organisms in every area, okay? Psychotrophs here are going to be our gold line, okay? And so you can see their optimum temperature is a little bit wider looking, and this is going to be from about 15 to about 30 degrees Celsius here for the psychotrophs. They grow slowly in the cold, but their optimum temperature is between 15 and 30 degrees Celsius. Now remember, this is Celsius, not Fahrenheit, okay? Staphylococcus aureus, listeria, these are able to grow at refrigerator temperatures, and so this is what we're going to see that cause our food-borne illnesses. Be sure you know that. You need to know these guys, their temperatures, where they are typically going to be problematic at, especially the ones that can make us sick. We really, that's our main focus, is the ones that can make us sick. So guys that grow well in the refrigerator, definitely need to know them, right? Okay, and so mesophiles, meso means middle, and so these are kind of the middle of the road guys. And so that's the green line there. This is the most medically significant area. This is where the majority of the most medically significant microorganisms are. They grow at an intermediate temperature between 20 to 40 degrees Celsius. They can grow from 10 to 50, but 20 to 40 is much more hospitable. Okay, these can inhabit animals and plants. They can also inhabit the soil and water in temperate, subtropical, and tropical regions. Okay, human pathogens have an optimal temperature between 30 and 40 degrees Celsius. So you can see why these are the ones we're going to be the most concerned with. Human body temperature, 37 degrees Celsius. Okay. So thermophiles are going to be our pink line. Thermo generally refers to heat. Okay. And so these guys really like heat. Heat loving would be what thermophile means. And they grow at temperatures greater than 45 degrees Celsius. They live in the soil and water in areas associated with things like volcanic activity, compost piles, because those are going to be warm, and areas that are directly exposed to the sun. Okay, and so they can range from 45 to 80. Most eukaryotic forms can't survive above 60 degrees Celsius. Now, we also have those extremophiles. So these are going to be our extreme thermophiles, okay? And we call those extremophiles, or those that live in these extreme areas like the polar ice caps. But here we're talking about the extreme thermophiles, and they're going to grow between 80 degrees Celsius and 121. So these are going to be those deep sea thermal vent type critters here. So we want to define two more words here. We've got facultative. That means they can grow under different conditions. So that means they like a specific thing, but they can grow under different. And then thermoduric, these can survive a short period of time being exposed to a high temperature. So Staphylococcus aureus grows best at body temperature, but it is a facultative psychotroph, so it can survive and multiply in the refrigerator. Okay, so this is this is a concern for food foliage when we're storing. Okay, Listeria is truly a psychotroph, and it grows best at 30 to 40 degrees Celsius, but it can still grow slowly at temperatures as low as 1 degrees Celsius. So this is why we had that problem with Listeria and ice cream. Okay, and so you probably remember that it also refrigerated meat. So that's something we want to pay attention to when we're looking at foodborne illnesses in those areas. Okay. Thermoduric can survive, like I said, those short exposure to a high temperature, but they're normally mesophiles. So this is a common contaminator of heated or pasteurized foods, because those were heated up for a short period of time, but probably not long enough to kill them. And so our representative bacterium here is Bacillus clostridium. Okay, one that can handle that. So we talked a little bit, or I mentioned that gases also can affect microbial growth. This is really pretty important. The main atmospheric gases that influence microbial growth are going to be oxygen and carbon dioxide. Oxygen's got the greatest impact on microbial growth. It's an important respiratory gas and a very, very powerful oxidizing agent. That means it strips electrons from things, so it can cause problems there. Microbes are going to fall into one of three categories regarding these. Those that use oxygen and are able to detoxify those toxic forms of oxygen. There are toxic forms of oxygen. Those are going to be those oxidizing agents. Those that can't use oxygen or detoxify it, and those that do not use oxygen, those that can use oxygen but are not able to detoxify it, and those that don't use oxygen but they can deal with those toxic forms. So let's talk about some of these toxic forms here. When oxygen enters a cellular reaction, it can sometimes be transformed into a toxic product here. Okay, so first here we've got singlet oxygen. This is just a normally oxygen is what we call a diatomic molecule. It is always combined with another atom of another type or another oxygen atom, and so that's why it says O2. It has to be there in pairs. And so when it's by itself, it's very, very reactive. It's very what we call electronegative. It wants to really pull electrons. Okay, and so it's extremely reactive. It can damage and destroy a cell because it can oxidize or pull those electrons from the membrane lipids. Okay, remember that membrane is that external boundary of the cell, so it's very important. One of the substances produced by phagocytes to kill invading bacteria is the singlet oxygen. Okay. And so, like I said, that can damage and destroy cells also. Superoxide is going to also be very reactive, and normally O2 is not going to have a charge. Well, this has a charge, so it's also very reactive, okay? We can also have, and it's going to act basically the same way as singlet oxygen. Hydrogen peroxide is also toxic. Now, you're probably thinking of hydrogen peroxide that we use in that on a daily basis, and that is, it's 3% is what household hydrogen peroxide is, but because it is toxic is why we actually use it to clean wounds. Of course, that small 3% we also use to help pull things out of a wound. Now, full strength hydrogen peroxide is extremely caustic and would actually peel away your skin. So while you could actually, I would not recommending it, but you could safely drink 3% hydrogen peroxide. Full strength hydrogen peroxide is really bad stuff. So yes, it is toxic to cells, and this is why we actually use it as a disinfectant, okay? But that's also why they've kind of moved away from using it to clean stuff, because it's also toxic to our cells, just not as bad as it is to something smaller like bacterium. And then lastly, we have this hydroxyl radical, that's OH negative. It is also very reactive. It is going to want to pull protons and hydrogens, okay? And so that's the four little main toxic forms of oxygen that can be produced. So a lot of cells have developed enzymes that can scavenge and neutralize these free radicals. That's what these actually are, is free radicals, these reactive oxygen byproducts, okay? And so to protect themselves against damage, they've got these enzymes that can convert things like your superoxide into oxygen gas, but you've got to have 2 steps in order to do this, okay? This is essential for an aerobic organism, all right? So superoxide is going to first be made into hydrogen peroxide, okay? So this is our superoxide here. You can tell because it's got that negative charge. Superoxide dismutase is an enzyme. If it ends in ASC, it's an enzyme that is going to help deal with superoxide. That's the great thing about enzymes. Their names tell you basically what they do almost all the time. Not always, but most of the time, okay? So the first step is going to be, we're going to make hydrogen peroxide, which is a little less damaging than superoxide over here. And we're going to make some normal oxygen gas, okay? And so we've discussed, however, that the hydrogen peroxide is still toxic, so then we're going to deal with that, and the way we deal with that is by catalase, okay? And so we're going to take catalase and convert hydrogen peroxide, break it down, we're going to catalyze it, we're going to break it down to water and oxygen, which is, of course, much better for the cell. So if a microbe can't do this, then that is when it's going to have to live in an oxygen-free environment, and it will be anaerobic. It's going to be an anaerobe. We classify our microbes by their ability to handle oxygen. So that's what we're going to look at here. Aerobes that can use oxygen, gaseous oxygen, in their metabolism and contain those enzymes that are needed to process those toxic oxygen products are going to be our aerobes, right? Okay, those are our aerobes. And so if it cannot grow without I'm tongue-tied today. Without oxygen, it is then going to be an obligate aerobe. It is obligated to live in an oxygenated environment. Things like protozoa, fungi, some bacteria like bacillus or mycobacterium, those have to have oxygen. Then we also have microaerophiles, and these guys need only a tiny bit of oxygen. They're harmed by a normal percentage of atmospheric oxygen, which is about 20%, 19-20%. But they do require some oxygen for their metabolism. So organisms that would fall into the microaerophile category are going to live in areas like the soil or water or a mammalian host, where they're not directly exposed to the atmosphere, but they still get some oxygen. So some examples here are going to be H. pylori, which is the guy that lives in the stomach and causes peptic ulcers, and Borrelia burgdorferi. And so when cultured, bacteria can be grown in something we call thioglycolate, and that allows anaerobic bacteria to grow in tubes exposed to air. Oxygen concentration would be highest at the top of the tube. And so the guys that really want oxygen are going to be at the top. If they don't like oxygen, they'll be at the very bottom. And then we'll have everything in between. And I believe, and hopefully we have a graphic of that you'll see where all these that we're talking about kind of fall. And so you can put them in this thioglycolate and classify them as aerobic or anaerobic. Are they in the middle? Are they microaerophiles? That type of thing. So we're kind of moving from the maximum amount of oxygen to the lowest amount of oxygen. So the third down on the list is going to be our facultative anaerobes. And these guys do not require oxygen, but they can use it if it is present. A lot of gram-negative intestinal type bacteria like staphylococci are going to fall into this category. Okay. Next, we're going to have our anaerobes, and these are going to be the guys that do not have the enzyme for using oxygen in respiration, which is our metabolic processes. Okay. That's going to be, again, some intestinal bacteria and a lot of the bacteria that can grow in your mouth. Below that, we're going to have, obligate anaerobes also lack the enzymes for processing that toxic oxygen as well, and they can die in its presence because they are not able to defend themselves against those toxic forms. All right. Aerotolerant, these do not use, and this is, I said these were in order, but these are not. Anaerobes would be at the very bottom. Aerotolerant anaerobes do not use oxygen, but they can survive and grow to a limited extent in its presence. They're not harmed by oxygen, but they, because they do have an alternative way for breaking down peroxide and superoxide. Guys that can do this are going to be lactobacillus, streptococci, some clostridial species as well. Although all microbes do require some carbon dioxide in their metabolism, capnophiles grow best with a high concentration of carbon dioxide. And so they really prefer more carbon dioxide, okay? Things like Neisseria, which is going to cause gonorrhea and meningitis, and those Neisseria species, Brucella species that causes the undulating fever, or Streptococcus pneumoniae. These are species that prefer to have more carbon dioxide. pH is a measurement of the degree of acidity or alkalinity or basicity of solutions. And so 7, of course, is going to be neutral. We're going to have on the 0 side is going to be the most acidic. So the lower the number, the higher the acid concentration. 14, the most basic. And we tend to say that is more alkaline when we talk about the body. Okay. So be careful with this. It's easy to mix up more acidic with a higher, you want to think a higher number. It's always going to be the lower number. Most organisms are going to be neutrophiles, meaning they prefer to be in a neutral environment here. About 6 to 8 on the pH scale is going to be where most want to live, and they're going to generally have a very low range for pH, so low tolerance and changes in pH, although we do have those on extremes. Like we do have some that are acidophiles, like our H. pylori. So here are some of our pH extremes. We have our acidophiles and our alkalinophiles. Okay, these are obligate acidophiles here. This Euglena mutabilis. Okay, and that is not all species of Euglena, because Euglena is often found in pond water. This is a special one that grows in an acid pool of a pH between 0 and 1, which is extremely acidic. An interesting one is this Picrophilus, okay, and that is one that is going to be a problem for spoilage of pickled foods, so hence the name, right? Then our alkalinophiles, Nitromonas, that's going to live in a hot pool and soil, and that's going to have a lot of minerals that give it that really alkaline pH there, because minerals can actually make things more acidic or more basic. H. pylori can actually produce ammonium, and that can raise the pH of solution, making it more basic and actually neutralize stomach acid, so it is able to live there. All right. And then Proteus can, or Proteus species, there's actually several species of Proteus that can create an alkaline condition and neutralize urine, and that so that they are able to infect the urinary system. And so many of these microbes can produce something to make their environment more hospitable for them to live in. So this is, by the way, going to be probably one of those longer lectures here. I try to break them up into 20 to 30 minutes. This one's looking like it's going to be a lot longer. I may actually split it up after I'm done recording this. So if that's not the case, that's why. All right, so osmotic pressure is another thing that can affect how microbes live. They live best under hypotonic or isotonic conditions, but there are some that can handle a high solute concentration, and we call those osmophiles. Okay, then we also have halophiles, and these guys fall under our extremophiles, and some of them do anyway. Halo means salt, and so these are salt loving. Okay, so a halophile is a type of osmophile. We have obligate halophiles. That's going to be our halobacterium and halococcus, which can actually inhabit salt lakes. So the great salt lakes like the Dead Sea, which we once thought nothing lived in, we have found out that there are obligate halophiles that can live in this area. Let's see. We then also have what we would call a facultative halophile, and so these are resistant to salt, even though they don't normally live in a salty environment. One of these guys is Staphylococcus aureus, okay? And that's important because Staphylococcus aureus is going to be in areas where we have salt concentrations in some of our bodily fluids.